Good afternoon. Let me first, like the other speakers, thank the foundation for letting me come here in recognition of King, who's been a friend and inspiration for, I guess, 34 years. I was trying to figure out the years. Like the other people, I want to say something about King, but I want to start much earlier. This is now, if you look at him today, and he's been seen by most of you, I never saw a cuter child. I never saw a child who looks more like the adult. It's kind of scary. And I never saw a guy more likely to go into STDs. So, I mean, if you were the parent that bore that child, you think, is he going to be a doctor, a lawyer, an Indian chief? You'd say, well, I'm pretty sure he's going to be an STD researcher. I'm fairly certain your parents probably said that to you over the course of your childhood, and so there was not much degrees of variation as you went along. Let's not focus too much on King. Let's go back to me. I don't want to get too turned away from my own narcissism. So I got concerned about this when I was in China, working for the university, actually for Yale at that time, and that's me with hair and much younger version of my wife, who's a China expert, is next to me, and those are our Chinese friends, many of whom we still collaborate with. And this is a daycare center where the Caucasian child's the one in the green coat, my very fat little daughter, and she'll come up in this conversation now and at the end of this discussion because it gives you a time frame. She was then a 1-year-old infant. The guy next to her in the Mao jacket, I don't think she's ever seen again. In the PLA, that's the People's Liberation Army uniform, which you could easily buy then. But I think she's never seen him again. But why do I show this? Because Mao was this tremendous force that had, complete control over China, and he did some terrible things, as I think everyone's aware, but he did some remarkable things in public health. And among the contributions he made in public health was this rare event where between 1950 and 1960, a country that was incredibly ravaged with STDs ultimately completely eliminated STDs, including syphilis, for about 30 years, roughly 30 years. And I won't go into all the ways they did this, but it was this remarkable achievement that would be impossible almost anywhere but in China. And so then when I was in China and subsequently writing this manuscript, it was certainly on my mind that it's possible to use drugs to make a positive contribution in reducing the transmission of disease. Certainly that's been our model in some other diseases as well. But then there's a lesson to be learned, and Tom Quinn, who'll speak after me, and I are trying to coordinate our talks. And so the lesson learned is so the Chinese virtually eliminated STDs, including syphilis, but then subsequently, as the country opened to the West, they've now suffered the worst syphilis epidemic probably in the history of the planet, with the most cases per 100,000 people on the planet. So there's some lesson in that as well, that history will repeat itself. And as I talk about the work that I've been doing, one has to be respectful of this kind of a history. And as I said, Tom will get into this as well. So in the end of the day, of course, the day has been, for the most part, devoted to classical sexually transmitted diseases, and now we switch to HIV. But of course, HIV is just another STD. But it attracted so much attention because of severe mortality and morbidity. In the absence of that, probably we would not be treating it the way we treat it. But yet it has this problem, and it has reached this massive proportion of 30 to 40 million people with HIV infection, and 80% of them in sub-Saharan Africa, and 80% of the HIV as a sexually transmitted disease. But it all began with just not a bang, but a whimper, just a few cases of people with unusual opportunistic infections recognized in Southern California, and that was just the beginning of this massive pandemic in 1980, just the time when I left China and moved to Chapel Hill, North Carolina. Now, by way of, how many of you have ever been to North Carolina? How many of you have ever been to Chapel Hill? All the Americans. But this is a very small town with just a few thousand people, and yet it suffered this massive epidemic of HIV, which really affected the career trajectory of many people in Chapel Hill. But Charlottesville, University of Virginia, which is just a couple of hours north, has no focus on HIV. And why is there such a difference between these two towns? What's special about Chapel Hill? Well, it turned out that Dr. Kenneth Brinkhouse, who helped to discover factor VIII, the clotting factor, moved to Chapel Hill. And in order for hemophiliacs to stay alive, they had to receive factor VIII. So Dr. Brinkhouse started manufacturing factor VIII, and thousands of hemophiliacs moved to Chapel Hill, and they bore little hemophiliacs. So by 1988 to 85, about a quarter of the hospital budget was for clotting factors. And as HIV contaminated these clotting factors, and by the way, at that time, instead of all the T-shirt shops, which every college town has, including this one, we had blood donation centers, which was a pretty weird thing. You know, instead of buying a T-shirt with UNC on it, you'd go give blood, because we needed a lot of factor VIII, and they were manufactured in Chapel Hill. And so this kind of contamination of the blood supply led us to take care of lots and lots of people. So by 1985, more than 10% of all the admissions to our 1,000-bed hospital were for HIV disease, and this was uniformly fatal. And so those of us who are old enough to have managed this problem in 85, it was really a devastating problem, to say the least. And then, in a rather one of the most miraculous moments in modern medicine, Burroughs Wellcome took a drug off the shelf, and investigators, shown in this paper, were able to show that this one drug reversed death from HIV, AZT, a simple one-drug regimen. Now, it wasn't perfect, but it really changed the world in which we lived in. It was certainly one of the major breakthroughs in this pandemic. Now, over the years, of course, subsequently, more than 30 drugs have been developed. The most recent one, dolutegravir, and these 30 drugs have really changed the face of HIV and taken HIV from a uniformly fatal disease to a chronic disease in which, if therapy is started promptly, early in the disease, the lifespan is probably virtually no different than the lifespan of somebody who's not HIV infected. So one needs to really respect this incredible contribution. Now, very early on, in '87, our group in Chapel Hill got involved in transmission and prevention of transmission. That was our goal. So I'm kind of jumping around in data, but with a very large investment, it was possible to understand the sexual transmission of HIV. And shown on this slide is really a simple idea, that people with HIV that's untreated have many, many copies of HIV, that's the virus growing in the blood, copies of HIV in their blood, and a similar number of copies in their genital secretions. We mostly studied semen. But if you have 10 or 100,000 copies, the virus, when it replicates, has so little fidelity that every copy is different. So there's 10 or 20 or 30,000 different viruses that might infect you. And in this very complicated series of studies, it was possible to show that when the transmission event occurs, that the swarm of virus is somehow sieved, and the transmission occurs from one virus or at the most 2 or 3 viruses. So the sieving event shows you the enemy, and increasingly we know that the virus, so the one virus is transmitted from hundreds of thousands of potential copies or tens of thousands of copies, and the one virus has some special properties. And understanding the one virus that's transmitted is in many ways the key to prevention and the key to vaccine development. We're not talking about vaccines today, but there's no doubt that that's an important field, and the amount of effort to make an HIV vaccine is undiminished. The other thing that happens, so the transmission event, so exposure occurs, and that's shown here. So here's the moment of exposure. If I can figure out how to work the laser pointer. These are very popular to blind airline pilots now, but that's their main use. They used to be used for, most of us don't point anymore because we have mice. We have mouse, but now these green pointers can apparently bring down planes, and so sooner or later they'll be regulating these. But so here's the exposure event, and a swarm of viruses at the mucosal surface, and one virus or 2 viruses are transmitted. And then there's a period when no virus can be seen. Why is that? And there's no antibody response, of course. The virus is only in the tissue, and it's trying to infect multiple cells in the tissues, but it probably all are eliminated except for this one replicating virus, and then then it ramps up and has ramp-up viremia. Now, ramp-up viremia is important for 3 reasons. First, that's the maximal contagion. So earlier this morning, Sevgi was talking about networks. And there's probably no one more important to a network than somebody who's introduced with earlier acute infection, because that virus has come from one person to another, and it's ready to go to the next person. It's got its bags packed. It's perfect to go to somebody else, and this maximal number of copies is achieved. The second idea is that during this window of ramp-up viremia, every compartment in the body is seeded, and the virus becomes integrated into the host DNA. And because it's integrated in the host DNA and every compartment is seeded, we cannot cure this infection. And yet, we are now desperately trying to cure the infection. So the challenge is to eradicate infection from every compartment, including the brain and the genital tract, and to figure out a way to get all those cells with integrated virus out of what's called latency. And we'll come back to that as I try and get through this talk. So acute infection is this critical moment in time, and I'll give a forewarning of where I'm going. It's likely that as many as a third of infections occur not from people with chronic infection, who have antibody and maybe even know they're infected sometimes. A third of infections are coming from people with acute infection that just got the infection, that proudly can say I'm antibody negative, but yet they can transmit very efficiently to the next person. Now, on this slide are all the ways you might prevent HIV infection. This is like one massive summary. And so we have lots of behavioral interventions, and one would not diminish the importance of treating STDs, delivering condoms, and use of circumcision, and Tom will talk a little bit more about this. Then at the moment of exposure, we'd like to have a vaccine that was providing antibodies that would protect from infection. We don't have that, at least yet. Tom will talk about giving antiretroviral drugs at the moment before exposure, or at the moment of exposure, that we'll call pre-exposure prophylaxis. But I want to focus on what happens if you treat the infected person. This brings us back to the whole syphilis argument. The whole way that the Chinese dealt with eradicating syphilis was, in fact, using single-dose benzathine penicillin massively to treat their way out of an epidemic, out of a pandemic. So in order to approach this problem, our group and many other groups started studying all the drugs. And this very unreadable, fatally complicated slide, there's a simple message. If you're above the line, it means that the concentration of the drug we're studying is greater in the genital tract than it is in the blood. If you're below the line, it means the drug isn't getting well into the genital tract. Why is this important to us? If we're going to treat our way out of this, the drugs that might most benefit your own personal health might not be the same best drugs to treat your way out of the epidemic, because we need drugs that concentrate in the genital tract and wipe out the swarm of virus in the genital tract. So for clinical plausibility, we need to look at these drugs above the line. Now, having spent about 10 years doing this, there was then enough biological plausibility that you could say, okay, these drugs get in the genital tract, they stop replication, it ought to be possible to stop the transmission of HIV from one person to another. So we developed a clinical trial that's a very simple idea. Could we treat people to reduce their contagion? That would be a transmission benefit above and beyond the clinical benefit. And then we would try and prevent transmission. And in order to test this hypothesis, the only credible way to do it was to find discordant couples. With respect to every HIV, everyone that's in a relationship is in one of three relationships. That both people are negative, and that's true of most people on the planet, so they're concordant HIV negative. Or both people are positive, that's concordant HIV positive. But we were going to have to find HIV discordant couples and to enroll them and treat the infected person to understand whether we could reduce transmission to the uninfected person. Now, this seems too intuitive. It seems like it's obvious. But we weren't really interested so much, did it work? We needed to know the magnitude. It's like a vaccine trial. If it only worked 20% of the time or 30% of the time, it wouldn't be very effective. So we needed to know the magnitude. We also needed to know the durability of this. Did it work for a month or a year or 5 years or 10 years? So we developed a clinical trial with the help of the NIH in which we did randomize people to immediate ART, where the index case had a CD4 count of 350 to 550, which would be high at the time we started. No country in the world was treating people at that level when we started. And let people, in the delayed arm, everyone was going to get ART, but they would delay their ART until the CD4 count fell to between 200 and 250, which was deemed as safe for the host. That is, the host health would not be compromised if there was a fall to that extent. At every visit, we had to be concerned about doing everything else possible to stop a transmission event. Tell everybody to use condoms at every moment in time. Have no sex with somebody who's not your monogamous partner. Counseling. And we spent a year developing a counseling script to try and reduce as much as possible transmission based on sexual behavior, risk behaviors. Now, that works against the goals of the study. If everybody used condoms 100% of the time, then the study would never go on. And the people told us every visit, because we were giving them free condoms and telling them to use condoms. They said, yes, I've used condoms 100% of the time. But we were pretty sure that wasn't quite correct because there were 234 pregnancies. So either there was massive immaculate conception, or people tell you there's a social desirability. And you say, here's your condoms, use condoms. And they say, yes, I use my condoms. I think this is a really important thing for clinical investigators to realize without biomarkers, and pregnancy is a wonderful biomarker because it's incontrovertible, I think. So that's the bottom line of that. So we were looking at linked transmission events. And when I say linked, we had to show that the virus from one person, that the virus from one person was going to the next person. It couldn't just be any virus, because if they had sex with somebody outside their primary relationship, we would be confused. So we had to develop some tools of viral linkage that we used. And in addition, we were looking at the health of the subject. Since we were treating people earlier, we needed to know whether treatment hurt them or helped them. So those are the two concomitant questions. Now I do not recommend this study. A lot of you are young, I could see. Everybody's younger than King, I'll say that 100% for sure. It took him a long time to get this award. I'm not sure, it really took you a long time. You were already in your 50s, and it took you 20 years longer, but, or 15 years longer. 25. He wants to give his age. I'm not going to give that. This study went on for 11 years with no abstracts, no publications, no reports of anything. So this was not a particularly popular study, and there were a lot of stages of the cross that prevented us from getting anything done, and they're all kind of listed here. But if you're starting your career, I do not recommend you do this study. This is not the way to have a successful career. But after 10 years, at the 10th oversight visit, because this was blinded to us, we went to the oversight visit. There's an oversight board of the Data Safety Monitoring Board, and they have the results. And at that visit, they usually told us nothing. At this particular visit, they said they have a recommendation. We said, what's the recommendation? They said, we can't tell you the recommendation, which we thought that was really bad news. And we were so blinded, we thought the study had failed completely. And they were telling us they wouldn't tell us the news because they were stopping our terribly failed, very expensive failed study. However, actually, and by the way, so this study was done at 1,763 couples were enrolled at 13 sites in 9 countries, 278 in the Americas, 531 in Asia, and 954 in Africa. We oversampled Africa because that's the epicenter of the epidemic. So on this 10th meeting, when they said they wouldn't tell us the recommendation, finally we learned that day, later that same day, what the recommendation was. They wanted us to make the results public. So they weren't stopping the study. They wanted us to tell people what they saw that we were blinded to. So what did they see? So they saw that there were 39 transmission events. For all of our counseling, 11 of those transmission events were unlinked. That means that the couples were not faithful to each other, and a third person got involved in the relationship, and that was kind of irrelevant to the goals of the study. Then we looked at the linked transmission. There were 27 in the delayed arm, where people weren't taking treatment, and 1 in the immediate arm, which accounts for then a 96.4% prevention over roughly 22 months of transmission ascribed to all the other things we did plus ART, because everybody got the same thing, and the ART was said to have led to this 96% prevention. So we showed that if people take their pills effectively, that the magnitude of the intervention is very, very high. Now, that one case, by the way, as you might guess, as obsessive a person like me, I was terribly upset about the one case. And so in PLOS 1 last month, we published that one case and showed that that person almost certainly had unprotected intercourse on the day we started the study. So that was a great relief to me because I wanted this to be 100% effective. So please look at PLOS 1 and realize how obsessive we are about this. Now, the other thing we looked at is the clinical benefit, and this is the 2 groups, immediate and delayed, and what you'll see is that the complication rate, if you don't get treated early, is greater. You're more likely, 50% more likely to get tuberculosis and more likely to get recurrent herpes and skin diseases. It's not a massive negative effect, but it's not good to delay your therapy. And a bigger issue for delayed therapy is on this slide. This is the immediate arm. Their CD4 count is going up, up, up very quickly. The delayed arm, the CD4 count falls. And when the CD4 count falls and you try and then treat people later and get the CD4 count up, it's much harder to get the CD4 count up. So there's this really big benefit to starting treatment early. So this became the breakthrough of the year, as was indicated, I think. No, you didn't indicate it, but this became the science breakthrough of the year. Please look at Bruce. This is my most expensive slide because his mouth moves. So I need to go backwards. So see this, the idea that I can get, that not me, that our group has, I don't think anybody's, King, you've never done this. You've never gotten a mouth to move. And so I would just go back and forth. So I'm very proud of that slide. So this became the science breakthrough of the year, and the runner-up of this was a Japanese 35-year study of a spaceship that went to an asteroid and collected solar dust that cost $2.7 billion. And we're 100% sure the Japanese were not happy that they were not on the cover, that the spaceship wasn't on the cover of Science. I'm sure that in Tokyo we're not popular. So then Tom will show this slide. This got construed as the end of AIDS, as an AIDS-free generation at the end of AIDS. And I think now we have to take pause and say, okay, this has a really powerful tool, but it's a tool, and Tom and I will both say a little bit more about the limitations of the tool, but Tom in particular will focus on that. What happened to 052? It's ongoing. Most of the cases and the controls are still in this, and all the controls were offered drugs, all but 73 have started ART. Now, why have the 73 not started ART? Because they've had 2 generations of messages saying you don't need antiviral therapy. The more that we've given that message, the more people incorporated the message and have tremendous reluctance in believing that we now know that it's better to start treatment earlier. So the study is ongoing for durability and magnitude and looking at the side effects of any delay in ART. We believe that everyone should start ART immediately. The U.S. guidelines now indicate, and I don't know about the Canadian guidelines, but the U.S. guidelines ask for immediate ART for all the reasons listed on this slide. There are some arguments about delaying ART, and I think they're, for the most part, falling apart. And I won't go into this in detail. There's a paper in The Lancet this week that has all of this in it about the arguments that surround starting immediately or delaying ART. And I would refer you to that paper. Now, the WHO has changed their guidelines to wanting to treat index cases of all couples immediately, wanting to treat everyone with CD4 count greater than 500 immediately, and I think that'll go to everyone eventually, and wanting to treat all pregnant women immediately forever. So when you look at that, if you have almost 36 million people infected, 26 million now are eligible for immediate ART. So this is a vanishing small argument about who should get ART, but the problem is we can't get the maximal benefit of ART until we have a uniform message. We saw this in hypertension. It was impossible to get people to benefit from lowering their blood pressure until everyone agreed that there was no modest hypertension. Hypertension was hypertension, and everyone with hypertension gets treated. So in the next generation, you'll see movement towards much broader use of ART immediately, and I think that will give the maximal benefit of these drugs. Rochelle Walensky, now if nothing else convinced you, so suppose so far you really don't believe there's any benefit in treating people earlier. Rochelle Walensky, my colleague at Harvard, has a paper this week in the New England Journal looking at all these results. It's next week in the New England Journal. And what she indicates is, for South Africa and India, is if you treat people immediately looking at transmission and clinical benefit, you actually save money. It's cost savings. If you treat and eventually, if you look at those 2 countries, it's termed as very cost-effective, which is the kind of thing that health economists like. In addition, there's a whole Journal of Health Affairs that has 3 articles devoted to different aspects of HIV intervention, and it indicates that when you treat people in developing countries with ART, the GDP goes up. Productivity goes up. So I think the use of these agents above and beyond the transmission benefit is compelling. So what about the population-level benefit? Some, but not all, ecological studies suggest that broader treatment reduces HIV incidence. What does that mean? If you treat a whole community, you stop seeing so many cases. That's a really powerful ecological argument for treatment. Now, Sevgi showed a study, and it shows an elephant. She showed one aspect of a study from South Africa by Tanzer, where she was focused on network structure, essentially. But what we're focused on more is what's on this slide. The green is no ART. The orange is more ART. They have the home address, and they're following 16,000 negative people in KwaZulu-Natal. And so they've got their home addresses, and they're measuring incidence every 6 months. And what they see is when you deliver ART to more than 40% of people living in a geographical space, that for every 1% increase of use of ART, there's a 2% fall of incident HIV. So this is a population-level argument that says, whoa, if you treat more people, you see tremendous benefit. Now, but is this enough for us to treat the whole planet? Probably not. So you see all these studies now starting, HPTN 071, CDC Botswana, ANRS Africa, and SEARCH. These are community randomized trials with some communities getting ART immediately, door to door, and others getting the standard of care basically. So on this next slide. This is the HPTN 071 study, a giant study in South Africa and Zambia, where you treat people door to door at 7 sites. You do better than that, better than the standard of care at 7 sites, and 7 sites have the standard of care touching about 2 million people, trying to show that this community that's touched by ART immediately benefits in GDP, health of the people, and reduced incidence of HIV. The success of these studies will then lead. Rob doesn't think they're going to work, but they might work, Rob. The success or failure of these studies, I'll let failure be possible too. The success of these studies will then further change these policies. But so why are we unsure? Well, there's tremendous logistical challenges, which Tom will talk about. There's increasing incidence in gay men, even though we're treating more and more people. Tom will talk about that. There's treatment optimism that really can offset some of the benefits, especially in gay men. And then there's acute infection, which we really don't know how to deal with. We can't find people in the first few days of infection. Our inability to find them leaves a huge pool of people that we can't treat. This is from a modeling study in Malawi that we're pretty sure is at least partly correct, where this is the best you can do in the absence of finding people with acute infection, and an elimination plan requires finding everybody with acute infection, and we don't know how to do that. So we might get 50, 60, 70% benefit, but until we deal with acute infection, we'll probably never be able to treat our way out of this, even if we dealt with all the other massive challenges Tom will talk about. So the last thing I want to say before I stop, and then I'll stop right on time, is there's only 3 things in this field. There's prevention, and I've talked about some of it, but not all of it, and the vaccine field would be a whole other hour discussion. There's treatment, and the treatment of HIV has been a massive success by any definition. And then there's curing HIV. That was not to be discussed until recently. While the cancer field has the race for the cure, the run for the cure, everything for the cure, the HIV field was almost blocked from discussing this as too aspirational. That time is gone, and so now we see massive funding to try and cure HIV. And on this slide are all the strategies. Get cells out of latency and eliminate the cells. And this drug varinistat, which is a particular drug that gets cells out of latency, followed by a monoclonal antibody, might have the potential to do this. There's a paper coming out in, I think it's PNAS, about mice that establish a benefit from this kick and kill strategy. The Argos dendritic cell vaccine. You'll see another paper from the Argos Corporation where they've taken dendritic cells, educated and put them back into humans, and show a benefit. There's therapeutic DNA vaccines that are being developed. There's cytotoxic T-cell therapy. There's the Sangamo gene therapy. Sangamo is a company whose stock is up and down, where they knock out a CCR5 receptor and argue that that is a way to eliminate HIV. And they're reporting, somebody told me at a meeting we were all at, that they have a report coming out in a prestigious journal about the first 12 patients, of whom 9 of whom had massive fall in HIV viral load, by using the Sangamo gene therapy approach. And then there's the Berlin patient, which most of you are aware about, and some Harvard transplant patients who were transplanted with routine transplant, but the immunotherapy required for the transplant seems to have knocked down the regrowth of the virus. So this is a tremendously active field, which has really great potential for the next generation. So in my mind, where we're at right now is we're at a bridging place in the HIV pandemic. This is 30 years of this pandemic, and this is what my daughter looked like when it started. This is what she looks like now, and she has her own 4 children. So it just shows this massive time that's gone by, a whole 2 or 3 generations, or however many generations it is, of work that have led to where we are today. And before 1987, there was no ART. Everyone died. In 1996, there was AZT alone, but most people could live. In 1996, we had triple drug therapy that became hugely important for people failing AZT. In 2006, this went from 20 pills a day to 1 pill a day, which was an earth-shaking thing for the field of HIV. In 2011, we published the 052 results, and Tom will talk about pre-exposure prophylaxis results that got people thinking more about antivirals for prevention. 2013, I didn't talk about injectable agents. We now have 2 agents that last several months once they're injected. And the the idea that we can treat people with 2 or 3 shots a year is now coming into real thinking. And I can promise you in 2015 things will change. This is not going like this forever. In the next generation of scientists, this disease will change dramatically, and I'm quite sure for the better. So I would say, here's the deal. ART improves health and it blocks HIV transmission. And so there's a real commitment to the idea of an AIDS-free generation, but that's only an aspiration. So as we go forward, we have to have humility, because we've been here before with treatment as prevention of syphilis or other diseases, and it didn't work out. But we have the most studied pathogen in the history of the species. And I think now is the time not to think, oh, things are going great in the HIV field. It's time to redouble the investment in virtually everything that we're doing. To focus on the most critical questions, not to do every single thing, but to look at how you get the most bang for the buck, and prepare for a long march. That's a Mao expression, because Mao walked all over China, 8,000 miles over China. This is going to take tremendous tenacity. There's a tendency in our species to believe you've succeeded and give up and stop. Now is the time to redouble all these efforts. And really, for the HIV pandemic, failure is absolutely not an option. You need to those who were alive in the polio era, and my first job was managing iron lungs. The polio people suffered a huge pandemic. They successfully made a vaccine, and they wiped out one of the worst pathogens on the planet that just dominated the lives of most of the people in my generation, and maybe King's generation. So I would say that we must redouble our efforts for HIV, be proud of what we've accomplished, and move forward. So congratulations to King. Thank you for all your inspiration over all these years and your friendship, and thank you for listening.